Hello, my name is Christina, and in this video, I will show you how Gideon can benefit veterinarians and veterinary science students. What Gideon stands for is Global Infectious Diseases and Epidemiology Online Network. We track every infectious disease in every country every single day, and out of those diseases that we track, quite a few of them are zoonotic. Let's see just how many. Here I'm using a fingerprint search to find all the zoonotic diseases in Gideon, of which there are 205. Now, suppose I wanted to ask a particular research question. Maybe I'm a student in the United States who is interested in all zoonotic diseases that are mosquito-borne. Gideon can produce a list of 15 such diseases, and from them I will pick West Nile fever as my disease of interest. Now, maybe I want to ask a question of what bacterial zoonotic diseases are found in the United States. And Gideon can produce a list of 37 such diseases. In here, diseases of interest are anthrax, brucellosis, and leptospirosis. Okay, now I'm going to change my search criteria a little bit. Maybe I'm just interested in zoonotic diseases that are found in dogs. And so I can have a look at this list, and giardiasis will be the one that I'm interested in. Now I can create such lists and download a PDF or share it with a friend straight from the interface. Let's go into the comparison table and see what I've produced here. So I added 5 diseases that affect humans and animals, and I can sort them by clinical findings on how they manifest in humans. But if I'm a veterinary student, what will be more interesting to me is this fingerprint comparison, where we can compare different agents, reservoirs, vectors, and vehicles. So right away, we can see that 3 of the diseases I chose are bacterial, 1 is viral, and 1 is parasitic disease. It is really good to develop that understanding on how these diseases are diverse. Now, suppose I wanted to look in more into leptospirosis. I can head straight into the disease note and have a look at the summary where reservoirs will be defined for me, also the primary causing agent and vehicles and so on. Also etymology, helping remember what the bacterium stands for. The images gallery could be useful for those working in microbiology classes. But what I might be most interested in is perhaps leptospirosis in the country of choice, such as United States. Okay, so leptospirosis in United States. I am in one of the 25,000 country notes that Gideon has on infectious diseases. And the first thing we see is this time and place write-up stating that leptospirosis is most common in southeastern states and Hawaii. So we can expect to see some information in regards to that. Let's scroll down. You can see the graphs and tables on cross-border infections. And here we have cases reported in Hawaii. Some state-level data here and reservoirs. Now, this is particularly interesting because you can see the data on canine leptospirosis, seasonality, raccoons, fox squirrels, sea otters, and so on. Research bits related to that, and everything is cited very nicely with peer-reviewed references is straight in the interface. And from here, we could go straight to the article if we wanted to. Now, looking at the prevalence surveys table, you can probably see many species of animals here. That's because Gideon has always supported One Health approach, and we continue to do so by collecting this animal-related data, not just on surveys, also on outbreaks, and I'll show you that in a second. Looking at seroprevalence surveys, again, we can see animal and human populations mixed in, and you can absolutely filter them and search for them with these little arrows. Now, in the notable outbreaks table, we have cases and deaths that relate to humans. But if I wanted to add animal cases and deaths, I can do so. And then I can filter by where the largest animal outbreaks have occurred. So for leptospirosis in the United States, that was in 2017 Oregon, where 304 animal cases occurred. And then in California in 2018, there was an outbreak among California sea lions. And here we have the Latin name for the species as well. Okay, let's switch it up a bit and choose a different disease. I can use this breadcrumb at the top. Instead of navigating all the way back, I can just select a different disease, and now I'm in the brucellosis in the United States node. The latest outbreak has occurred in 2023. We have a little bit of a note here about time and place, swine brucellosis, any occupational exposures. So every disease will have disease-specific notes as well as the standard sections. With brucellosis, we have a brucellosis in animals section. Now, Brucella abortus, Brucella suis, Brucella canis are known pathogens to infect animals, and so we also track data related to those. Okay, now what can we do with all this data? We can visualize it. We can have a look at the outbreaks map of brucellosis, for example, and identify where the hotspots are. So in here, we see annual disease rates per 100,000 population and different levels of endemicity. We can see that brucellosis is most endemic to China, Turkey, well, Middle East and Southeast Asia. We can zoom in if we like and see particular places and cases and the dates or outbreaks that have occurred. Okay, another thing we can do, we can visualize this data in a different way. For instance, we can create bespoke graphs. And this is a really powerful tool because it can save weeks of research time of collecting this data, cleaning it, and putting it together. At the moment of speaking, we have 49,000 graphs. That number is continuously growing. Suppose we're interested in animal cases of rabies in the United States. See how I searched by keyword to create an AND statement and list all the graphs that may be of relevance to me. So I would like to find out animal cases of rabies in Pennsylvania and surrounding states. So I will start going down the list and selecting the states that are nearby, such as New Jersey, New York. Here's Pennsylvania itself as well. And now I can generate a chart, and we can see that New York is actually in the lead. I can hover over any data point here, and Gideon will expand the data for me. I can export this graph in a nice picture format for my presentation by clicking this download button below, or I can view the data in a data table and also export it, whichever one is preferable. Okay. We visualize data. Now, what else can we do? As veterinary scientists, we might want to identify the pathogen itself and learn more about the microbe causing the disease. So for microbiology classes, we could go into probability identification. There are two ways to do this. Decision tree is perhaps more fun and engaging, and it looks like so. You can generate various dichotomous keys by using this tool. But what I'll show you today, I'll show you the probability engine. So I'm in a probability engine. What's going to happen on the right-hand side is Gideon will calculate Bayesian probability. It will do the Bayesian analysis based on the phenotypic characteristics that I'm entering over here. And so I have a pathogen that happens to be gram-negative bacillus. It is not acid-fast, non-motile. It is an aerob. It is also capnophilic, and it grows at 42 degrees Celsius. So you can see how the pathogen names are switching places on the right-hand side now, and Gideon is calculating the probability. Because Gideon knows how common is each phenotypic characteristic in each isolate, it can do the sort of calculation to help narrow down the list of what pathogen you might be dealing with in the lab. So I'll do a couple more tests. Oxidase, catalase, positive. And also urea hydrolysis positive. And now we narrow down to 4 pathogens. And the top culprit happens to be Brucella abortus. It's a well-known pathogen in the veterinarian world that causes abortions in cattle. We can question Gideon. We can ask why something did not appear on the list. For instance, Why Leptospira is not on the list? Well, it's because these phenotypic characteristics do not match what Leptospira usually looks like. And I can have a look at the differences by adding this to comparison list. I can also add Brucella abortus to comparison list. And I'm going to go ahead into the bacteria comparison table and see how they are different. So we are showing only distinguishing characteristics here. What is always present in Leptospira? What's never present in Brucella? How are they different here? Or we can go ahead and compare entire phenotypes, if we like, side by side. Another thing we can do, we can go into the name itself, click on the link and open up the micro profile. And here we will have the summary, the etymology explaining the meaning behind the name, notes, ecology, any drug susceptibility. So we know that some antibiotics are used in both humans and animals, and this could be a useful place to find such things. Images gallery and the whole phenotype spelled out, which we can now email to a colleague or peer that we're working with, download the PDF, or view the references in a single place. Okay, well, I hope you find this short video useful. Thank you very much for watching.